Yeah, that's a little trail. Yeah, right, more. Yeah. Good, probably this is.
Yeah, we can kick it off here at noon. And we got a pretty,
On the turns out. Yeah.
all right, we got a pretty full house here in person, which is awesome. So thanks everybody for coming in person and everybody online. As always, we'll start with announcements. I have a couple. There's a new Kernza product on the market. So if you know Kodiak cakes, you can now get Kernza Kodiak cakes. I had them described to me as very dense and a little bit like sandpaper, but apparently, apparently that's Kodiak cakes, not Kernza. Kodiak cakes are notoriously very dense, and that's the thing. So it's not a knock on Kernza. So
Kernza was kind of lightening them up.
that's right.
Yeah.
Keeping it smoother.
Where can I buy that, Mitch?
You can buy it online. I don't know about in store. There might be some in store too. but- You found that in Walmart. Oh, great. So now there's two Kernza products in Walmart because there's also the Kernza Cascadian Farm Flower Blight in Walmart as well. So
Well,
that's exciting.
I know,
So everyone knows, we've been in conversations with a number of folks in Walmart, in the Walmart Foundation, and there is a strong interest in renewable, regenerative agriculture, and they are looking at the Forever Green program as a contributor to the products that they would like to put into, believe it or not, Walmart.
I don't know, Colin, are you online right now? I know you had a very busy day. We have Colin. All right, well, I'm just gonna say what Colin said to me, which was apparently a number of 3 years ago, talking to the Walton or Walmart folks, they said, well, how long is it gonna be before Kernza is on a Walmart shelf? And Colin did the math, and the answer was 1,000 days. Like 1,000 days from that engagement to that flower, and now I guess the Kodiak cakes being on Walmart shelf, so that's pretty cool. Other announcement is that the legacy bill, which includes the clean water legacy money, has passed the Senate and House legacy committees. Both of those committees included $6 million for Forever Green, the most ever. And the broader bills are pretty much identical, if close to, if not identical, which is good because it means there'll be less horse trading and shuffling. So there's a couple more steps. Yeah, they're first the bills go to the finance committees, then they go to the whole floor. So there's more steps to go, but still looking good for that fundamental Forever Green research fund. Any other announcements from the Forever
Well,
Green?
we had a great tour, and we announced that it's at the last week. Maybe we already talked about that the last time.
I think we did talk about that.
Yeah, but just to remind everyone, it went really well. And then we'll have 3 additional tours that are being scheduled with various groups from the St. Paul Garden Club to the Clean Water Council to the Department of Agriculture's Clean Water
Oh, yeah,
program.
their water quality certification program.
Certification program.
Yeah, and others, I've got others in my inbox that I haven't even told you about, Don, so we've got a busy schedule.
Is the governor part of all those?
We don't know, we haven't tried. We don't know.
There were requests out to the governor, to the House Ag Committee, since the last tour was Senate Ag Committee members, so yeah, there's more potential out there. Anything else before we turn it over? All right, take it away, Deb.
All right, well, thank you. It's fun to be here today and to talk about what we always call the first Forever Green crop in Minnesota, which is alfalfa. And so we rebuilt this program over the last few years, and so I just wanted to remind you who the ARS people are here on campus that work on alfalfa. So I have the research leader position, and so I also do plant pathology and biotechnology. We then hired Josh Gamble as an agroecologist, and he'll be talking today. Zhanyou was next as an alfalfa geneticist. And then we hired Jill Hoishley in forage biochemistry. And she'll be talking today. Dan Schlatter in soil microbiology. He'll also be talking. And then Sean Curtin has a portion of his time in alfalfa where he works on alfalfa gene editing. But a lot of his work is also focused on soybean. So we're currently rewriting our 5-year plan. And so we are interested in increasing the footprint of perennial forages on the landscape. And our project is quite broad, as you can see from the people who are involved in it. Everything from increasing alfalfa yields, improving the forage nutritive value, looking at increasing stress resistance, both biotic and abiotic resistance, enhancing environmental services, including soil carbon accrual, and then developing new uses and novel breeding methods. And so I will talk a little bit about what we're trying to do in some novel uses. So this was always my favorite exam question for students, to ask them what are the main crops grown in Minnesota. And they always get corn and soybean right. And then they kind of pause as what's number 3. And sometimes they'll say potatoes or wheat, or they're really stumbling, but it's actually alfalfa. So depending on how many wheat acres are put in each year, alfalfa is the 3rd most widely planted crop in Minnesota. And across the country, it's either number 3 or 4. But ever since I started here, there's been a concern that the acres of alfalfa are declining. There's been a kind of a shift. For a while, there were increasing acres in the West as the dairies moved west, and declining acres in the Midwest. But now there's really a decline across the country. Not the only place where we're seeing alfalfa acres increasing are in the Dakotas, both North Dakota and South Dakota. And so there's a lot of reasons for the decline in alfalfa acres. And so it's very complicated, but it's been a shift toward row crops, primarily corn and soybean, as this group well knows. Also, the dairy and beef numbers have declined, particularly in the Midwest, although milk production has been stable or actually increasing as we've gotten more milk per cow. But corn silage has been replacing alfalfa in the dairy diet. And some of that's because just ease of production. Farmers feel like they get more tonnage per acre. And then also the dairy nutritionists have not been on our side. They have been developing diets that are cheap. And they've not been inclined to add alfalfa into the dairy diet. And so we're trying to reverse that trend somewhat, because it's actually a really good feed for dairy cattle. It really promotes a lot of health and productivity.
So it's basically this cost, you're saying, that the reason they have left alfalfa out?
I think so. And part of it is because the variability. So the crop can vary depending on what harvest it is, on environmental conditions, and they want something that they can just plug the numbers in, and they don't have to change things too much. So based on the amount of protein or digestible fiber in. So what we'd like to do is come up with new uses that would take a lot of acreage. And we really have a lot of new acres of alfalfa, and it would also improve farmers' bottom line. So we've looked at, can we refine alfalfa? So there's all of the other crops. You can think of corn and soybean. They're processed into hundreds of different products. And alfalfa is basically processed into meat and milk. We don't process it into other consumable products. So there's been a lot of work on alfalfa protein as a nutrient concentrate going back to the 70s and 80s, although it never really quite caught on. It was called Prozan because it's protein and xanthophylls. There's been some work, and Joe and Zhanyou are working on some of this, and looking at some of the nutraceutical part of capacity in alfalfa, that as we start breaking it apart to extract protein, we can extract some of these other things. And then, of course, you're always going to have a fiber fraction that could be used in animal feed and potentially bioenergy. So some of the work that both Zhanyou and Josh and Sean are doing is trying to modify alfalfa to help reclaim phosphorus in the soil where we've added a lot of phosphorus through manure application. Potentially use alfalfa also in water buffers, protect streams and lakes. And then Josh has been doing a lot on soil carbon credits. We're pretty excited about the potential for alfalfa and human food products. It does the primary protein in alfalfa is Rubisco, which has a really, according to food scientists, it has tremendous capacity for being processed into fake meat, into meat crumbles, as well as egg white substitutes. So it's very amenable to human food products. We just need to get people interested in it. So some of the things, some of the bottlenecks to this is that it's always been, in contrast to grain crops that you can harvest and store, to get this alfalfa protein, it always seemed to be a really difficult process. That you have to harvest fresh material, because once you start drying the material, as Joe has shown, the protein starts breaking down. And so there have been machines developed recently to separate the leaves, which have the most of the protein, from the stems. But still, then you have to juice those leaves that are degrading fairly quickly. And then you have this juice, and you have to go through this whole process of heating it to coagulate the proteins and centrifuging it and drying. So it seemed like a lot of work, so that's kind of a big bottleneck to this alfalfa protein product. About 2 months ago, I got a call out of the blue from a guy who had worked in the 1980s in developing what they called a probine, a combine that would take fresh alfalfa from at the beginning of the machine and produce dried protein crumbles and a fiber fraction out the back. So a combine for this protein. They worked for International Harvester, and they actually developed this probine. They had a sort of a smaller model, and they had a full working model. And then International Harvester went out of business.
I actually saw the piece of equipment. It was sitting, 3 of them were sitting right over here at International Harvester on the corner of 280 and University. That's where they operated out. And they actually tried to hire our department head to lead that program.
Oh, I had no idea.
Yeah.
So the tech, and they patented it. They have all the drawings.
Yep.
It can be done.
And
So,
the person that talked to you owns those drawings.
yeah, he does. So he did try to, his name is Bob Alverson. He did try to restart this up again with New Holland in the 90s. I think they didn't have the funding to do it. But they tested the protein product at General Foods, which also went out of business. And they tested the fiber fraction at UW River Falls and Silage. So all this, but none of that data still exists anywhere. It's all gone. Then they were starting to look at the effects of soil compaction, because this would be a heavy machine going through the field. So you'd probably want to have, dedicated lanes for driving this. But so that was pretty exciting, so that we could actually, get rid of some of the bottlenecks to this protein production. So the other updates is that in this current budget, there was $1 million, 900,000, ARS takes 10% off the top, for recurring annual recurring funds for new uses in alfalfa. They're in the process of putting 2 new scientist positions in the functional foods research unit in Peoria. And so this is a unit that has a long history of developing food products and has ties to industry. And so they'll be working on extraction methods and how to utilize the protein food products. There's also a new scientist at the Dairy Forage Research Center in Madison focusing on alfalfa protein. And as I mentioned, Joe and Zhanyou are working on alfalfa protein from the breeding and nutritional component aspects. I'll also mention briefly, you saw this on the tour if you were there, that we have this cute alfalfa plant called Ruby. That the Ruby marker was originally just to track transgenic plants. But we found that alfalfa really makes a lot of this betaine, which is the red pigment in beets. And so this could potentially be sort of a new source for the super beets. Because the product that's made, even though it's made in a transgenic plant, produces exactly what you get, pretty much what you get out of beets. And Joe is looking into how this may actually help dry matter digestion and milk production. There were some studies out of China where they showed that they added this betaine to animal feeds and increased both meat and dry matter digestion. And it's also potentially used for foods and cosmetics, dyes. But we're not going to grow, hundreds of thousands of acres of ruby. This would be a fairly small amount. And I've talked in the past about our projects to use alfalfa protein in aquaculture feeds. And we've done 2 studies now, one here on campus looking at yellow perch, and yellow perch will accept the feed where we replace fish meal with the alfalfa protein. And then we currently have a project with people at UW Milwaukee where they've replaced the soybean in trout feed with the alfalfa protein concentrate, and have shown this to also be very effective, and have trout on-farm trials this coming up this summer to utilize it on a larger scale. But it all depends on being able to produce this protein concentrate in amounts high enough that you can get industry interested in it. So this is maybe something discussion after we've all finished is, for this group, you have a lot of experience in developing market demand. So we would like to get, your input on how do we create this market demand for alfalfa protein. And I'm looking at putting together a NIFA grant proposal where we get people together to develop a framework for this supply chain, where we look at everything from the breeders to the agribusiness people. And so any kind of input this group can give us on doing that would be much appreciated. I'm going to stop there and let some of my other folks talk a little bit. So next up we had Joe Hoishley, and you want to come here and advance the slides?
She wants to drive.
I want to drive it. Yep.
It'll make things easier. Thank you for having me today. I want to talk about digestibility. And in that case, I'm going to talk about animal digestibility. We need to think about who our consumer is. And here in Forever Green, you guys have some crops that you may use as a livestock stream as a source of food. But not all forage digestibility is the same. You can be a cow, a big ruminant. You're very different from a small ruminant, and the different industries require different nutrient intake. And of course, your customer might also be a human who doesn't understand that there are these fine lines, and all they see is an RFQ value. So there is an antagonistic relationship between quality and quantity. This is a just very excuse me, going too fast. This is a very standard picture that shows us that as the plant ages, it gets bigger. But at the same time, as the plant ages, the quality of the forage goes down. And the farmers are trying to find that sweet spot and what is the best way and when to harvest. But it also kind of talks about what kind of tissues. To get everyone on the same page, I'm going to talk about all the different characteristics here that are components that are involved in digestibility. So in alfalfa, our stems are highly fibrous, and therefore your non-digestible fiber, ADF, is really high with your crude protein really low. Leaves, easily digestible, have lots of nutrients in them, so it is the reverse. My research studies focuses on the stem because that's the limiting resource. if we can increase the digestibility of the stem, then we're increasing the digestibility of the whole plant. And if you zoom in and look at a cell, the cell solutes are 100% digestible if the animal can get to them. Then you have the cell wall. About 20 to 60% of that cell wall is digestible. So when you look at the cell overall, you have about 25 to 35% of non-structural carbohydrates, your sugars, your starch, and your pectin. You have about 50% structural carbohydrates, your cellulose, hemicellulose, and lignin. You have protein and lipids in lower concentrations. If you are doing a study on digestibility, you may be familiar with the terms NDF and ADF. NDF is neutral detergent fibers. Generally, these are highly digestible. ADF is acid detergent fibers. Generally, these are indigestible. Some people use these as placeholders for hemicellulose and cellulose. These are not plant traits. These are digestibility traits. It's important to recognize what you're measuring based on your assay.
You're looking at me when you say that.
I'm not.
I always do that.
Because digestibility is dealing with not only it deals with the interaction between three kingdoms, not three species, three kingdoms. You've got the plant kingdom, you have the animal kingdom, and you have microbes that are within that gut. So digestibility is the interaction between all of them. And if you're breeding for something, the ideal trait you want is actually a trait that's found in your organism, plant trait. So my goal with the study was to kind of determine what better plant trait is there for predicting digestibility, looking at cell walls. This experiment was set up by my predecessor, Dr. Hans Young, and the former breeder, Joanne Lamb. And what they did is they took 6 different populations or varieties of alfalfa, and because alfalfa is open pollinated, they mixed them all together and bred them. They got over 2,500 seeds. They put those seeds out in a common garden. They grew them all up, and then they measured all of those individual plants for either 16-hour digestibility, which gives us a number for how fast the alfalfa will be digested in a cow, and they digested it with a 96-hour time point, which tells you how long will that tissue persist. And they got some plants that were really high in one and low in the other, or they're high in both or low in both. And so what they did is they kind of they plotted it all out on a graph, and they drew an arbitrary cross, and they made selections in each one of those quadrants. Okay. Then from that selection, they grew all of the population of that group, once again making a selection from that particular quadrant. So if it was high, then individuals from that high quadrant were moved to the next cycle. Okay, and you ended up with 2 cycles of selection. We did two years of growth, and you ended up with 1,795 data points. For each one of those data points, they had digestibility, 16-hour and 96-hour. They had sequential fibers, ADL, ADF, and NDF. And then they looked at true plant traits of the cellulose, hemicellulose, lignin, and pectin. And when you look overall the comparison, sure enough, as the plants mature, the digestibility goes down, whether you're looking how rapid that digestibility is, or whether you're looking at overall potential of digestibility. And this is kind of correlated with, as the plants get older, their cell wall components actually increase. So we did some correlations with digestibility and all of these different traits. And as you can see, they're all highly correlated, or relatively highly correlated. And I'm very interested in how things are put together. So I did a path analysis. And the thing I want you to take home from this particular path analysis is that in 16-hour digestibility, the most important compound was cellulose here. For every 1 gram to kilogram of cellulose, you increased, digestibility decreased by 0.72. Okay. This was not what we expected. We expected to see something similar to the 96-hour one, where lignin, you reduce the lignin, you increase the digestibility. Okay. Why do I say that was expected? Because there has been a movement in reducing and creating low lignin lines to improve digestibility. Well, according to this data, sure enough, the overall potential of digestibility is increased if you decrease the lignin concentration. But alfalfa or any forage stays in the cow's rumen longer than 48 hours. So what you're actually looking for to increase digestibility is what's happening at the rapid time point. And this data shows that we should be looking at cellulose.
And Joe, quick question too, kind of thinking about like recent increases in dairy productivity too, right? It's really about the volume of forage, or one of the main things. They're just moving more forage through animals now. So
Yes.
it's getting shorter and shorter, from that 48-hour time point is my understanding.
That is correct.
Yeah.
So these are looking graphs. I'm sorry that they're really small. But what I'm trying to show you is that the red line is this high by high digestibility selection. The blue line is the low digestibility selection. Green is the high by low, and the black is the low by low. Those are the colors for the rest of these graphs. But you can see that both for the 16-hour and the 96-hour digestibility, if you choose a plant in the high by high quadrant, over time, you increase. If you select for low digestibility, over time, you decrease. And this appears to be linked to the reverse relationship you see over here, which is your cell wall components.
How do you select the microbes for your digestions?
That's a very good question.
Yeah.
We don't select our microbes. What we actually,
What
what do we do is
Yeah.
there are cows down here at the dairy that are fisculated. They have a hole in their stomach. So you put your you go through the fiber mat and you pull out basically fluid and stuff from their room and you squeeze that out.
So it's a direct culture.
It's a direct culture. There has been research on trying to create a single culture of I guess man-made culture. There was one that was sort of correlated with natural rumen, but the chemistry was lost.
Yeah, I don't want to but the engineering of the community, like for human consumption, when you can take out the ruminant and the ruminant part of it, that could be an avenue.
So but you bring up a good point. When we do digestibility, these in vitro digestibilities, you have to make sure you're using the same cow because each individual is different. And you have to make sure you use the same time of day because the daytime will what do they have for breakfast makes everything a little bit more complicated. All right. So the cell wall components, when we look at cell wall components, you can see here that the red and the blue are our extreme populations, and then we have our middling populations. And here, the concentration of lignin decreases with your increased digestibility, which is what we expected. And we noticed that following the black line, that's high 96, low 16. So it just further supports that lignin is driving that relationship for the 96-hour, the potential. And then here is glucose is also the way we measure cellulose. Here, there's really no change between our extreme populations, but you have an increase in cellulose when you're looking over potential of digestibility. And this is the 16-hour, you have an increase of cellulose. Yeah, an increase of cellulose when you have increased digestibility, which is good, actually, because cellulose is the building block for the food, for the livestock. And then a decrease there for the low-high population, which is following low digestibility. All right, hemicellulose. If you increase hemicellulose, you decrease digestibility. Pectin, we want more pectin, high pectin levels, better digestibility. But I broke these up. Sorry about all the slides. We broke this up, looking at the components of hemicellulose with the idea that maybe there's something going on. And instead of looking at hemicellulose as a whole, maybe there's one sugar that we can easily control to change. And for instance, xylose is highly indigestible. So if we could reduce xylose but increase fructose, then we now make hemicellulose more digestible. Same thing with pectin. If we uronic acid is not very digestible, but galactose and arabinose are. So what if we just switched what is the components of pectin to increase digestibility? So in summary, there's really no difference between the relationship between the populations, which is good, which means breeding has inadvertently changed relationships. There are changes in cellulose that can increase the rate of digestibility for rapid time points. The changing of lignin is increasing, can increase or decrease the potential overall of digestibility, and that there are changes in specific components of the cell wall that we might be able to also kind of address to impact overall digestibility. And for future work, we're looking at genes to look at the carbohydrate cellulose construction, and then also looking at maybe if we can allocate lignin to some other area so that you only have certain parts of the cell, the stem's cross section to have lignin in it, and everything else have available cellulose, that would also increase stem digestibility. All right. Any questions? Are we waiting for whole questions? Yeah.
How does like altering these cell wall components impact like methane production out of these ruminants? Or do you not know?
We do not know, but that is something to look at.
Cool.
Do you have a sense of how far you push those traits before you end up with like a plant that can't stand up straight or something like that?
Not as of yet. So in my previous life here, I worked with the oats, and we constructed a engineering model of the stem of an oat and barley. And we found some specific traits in that model that you can manipulate. The nice thing about the model is you can manipulate it out of the computer and realize where the edge of being too far is before you impact the structure and integrity of the plant. And we're doing that now, right now in the planning stage of doing that with alfalfa.
Maybe a bit related. So what kinds of genes are you targeting? And would it be with like CRISPR? Would it be transcription factor type genes then?
We really haven't gone into great depth about that particular area.
Okay.
Because I enjoy the physics more.
But yeah, all these have been genotyped, and so we're just waiting to get the everything that she talked about in the field. So we're just waiting to get the phenotyping completed, and so we can do some marker trait association.
Oh, okay. So you're not trying to create novel alleles. You're using the alleles that you already have.
Right, yeah, so the selection did increase digestibility and decrease lignin, but didn't change the leaf to stem ratio. So Joe is going to be doing some microscopy to try to find out, where in the cells the changes really occurred.
Gotcha.
Have you looked at year-to-year variation, like our year 2 plants? Do they have more cellulose? Maybe year 3 plants are even tougher to digest, or just primarily year 1 plants?
We did multiple year to year, and what we found is actually they're pretty stable once they mature. It's more important to harvest your plants, whether it is at an early bud stage versus like a green pod stage, like a mature plant. And once you cut it back, you're almost starting back from zero with that plant material. Now, yeah, for digestibility, that's what you're looking
The stand age is
at.
pretty much.
Stand age is not driving.
Dr. Cohen.
So, Joe, there's a couple of groups like Joe Chappelle and Ron Sederoff who created trees that are lignin minus. They grow fine, but they look like trees. And they've done that mostly for the bioenergy idea that you would produce fermentable cellulose. But have you thought about using their technologies to produce zero lignin? Is there a reason why you wouldn't want a zero lignin alfalfa?
My main concern would be lodging.
Why? Well, no, that's not, well, you can have a whole tree. A pine tree doesn't fall over.
Right, but with the transgenic low lignin alfalfa that has been developed, we do see morphological changes in the plants themselves. Deb, you were gonna say something?
Oh, I was just say, the Harv extra material that was decreased lignin, some of their earlier events did lodge terribly if they were,
Well,
decreased the lignin.
That's true in trees as well, but that's not when you specifically target individual aspects of lignin production. Mainly because when you breed for it, you get pleiotropic effects. And so anyway, just something to look at is actually look at the kind of stuff. I mean, Ron Sederoff is crazy, but he's actually a very good scientist. And but they actually have done quite a lot. And surprise, I thought if you didn't have lignin in a tree, it wouldn't produce anything. It would just kind of fall over and be a limp pine tree. But that's not what you get. It turns out that a lot of the rigidity is actually cellulosic, and so even though secondary cell wall formation does make better wood, it doesn't necessarily cause you to have those effects. So it's just something to look at, because some of the breeding programs actually give you a mischaracterization because they only look at one readout of the process. I mean, they're looking at the genetic selection, but they're not looking at the entire metabolic consequence of their breeding efforts. So just something to consider in that process. I did, Debbie, send you a link to a paper which uses Rubisco as a supplement to wheat to actually improve the nutritional quality of different uses of wheat, because as an additive, it actually greatly improves the nutritional quality without affecting the baking qualities of wheat. And it's kind of an interesting use of Rubisco. And I think that, that one of the things you get a lot of out of alfalfa when you squeeze it is lots of Rubisco. So anyway, I just thought I'd add that because I thought some of the work on trees is relevant to work on all plants.
Yeah, that's a good idea. So is that tree breeding, is the traditional breeding was gene editing, was gene engineer, the traditional
Yeah,
GM?
no, it's GM because they specifically knocked out specific steps in liquid biosynthesis.
Okay.
Yeah.
Yeah, well, that's good to know. And we can try, since Sean can, if Sean can do some gene editing work target to get zero lignin alfalfa, maybe good idea. So the one, well, Sean is here. So Sean, have you read that paper, Zero Lignin Tree?
No, but I'd be interested in reading it. So yeah.
Well, I mean, there must be a lot of papers. I don't know. I'm not an expert, but Ron Sederoff's a member of the National Academy, so this stuff must be around somewhere.
Yeah. Well, the program Josh presented is a complete conventional breeding, and no even marker system. Everything is conventional. So, yeah. Good idea.
Yeah, you have another question here.
I mean, back to the methane conversation, do you have a way to measure the methane coming out of plants?
What we do, so my lab, we measure in vitro. So that's grabbing the rumen out, and then we have a gas production system. And so you can totally get gas production, and then we take off, lead off some gas constitutive to our GC to measure methane, hydrogen and CO2. Then there are developments or technology that allows you to actually measure it from a cow. There's a green feed system where the cow actually puts its head in the chamber as it's eating, and then you get methane measurements that way. There's also USDA has a gas chamber. Yeah. But it's a room that the cow goes into, and everything that's emitted by the cow is then measured. And the cows can only spend so much time in that because they're actually removing system. But yes, there are many ways we measure that.
Oh, cool. Thank you.
All right,
Josh, take it away.
switch gears a little bit, talk about things more at the agro-ecosystem scale. So my work, when I say forage agro-ecosystems, I'd characterize that as spanning from questions about basic agronomy. How do we manage alfalfa and other perennial forages for more, traditional outcomes, yield, productivity, then to scaling up to understand how those management decisions impact things at the agro-ecosystem scale. So the structure and function of our agricultural systems, trying to optimize for a broader suite of outcomes, right? Focused on ecosystem services. And so I guess for those of you who attend these meetings regularly, you probably saw the presentation last fall. We've got a large collaborative project with Jake Youngers, Jess Gutnick, and we've got grad student Jake Kunder, who's doing a great job on that as well. We're also doing some work with C Yang now. But this is a project that's focused on quantifying field scale yields, carbon balances, greenhouse gas flux from sort of two different models for continuous living cover. So trying to integrate forever green crops into field scale rotations and understanding the impacts on yield and productivity, but then also on outcomes like carbon balance and greenhouse gas flux. And so we've got one system that's more of a perennial model. We've got alfalfa intermediate wheatgrass rotation, and then another field that we're doing more of a summer annual grains with winter oil seeds. We've been working a lot with the camelina team on that, integrating winter camelina into that. So thinking about different models for continuous living cover. And then trying to better understand sort of the spatial variability. So the other benefit of doing things at the field scales, we can look at some of the variability that we see, so that we can capture things that we don't necessarily capture at the small plot scale. We've got another project that's starting up this fall. That's a collaboration with Allison Duff. This is another Flux project where we're specifically focusing on alfalfa. And we'll be looking at alfalfa grass mixtures versus monocultures instead in terms of field scale carbon and greenhouse gas fluxes. We're also working right now to get at actually just this question about emissions. We're working with a group, Joe, and a group from the Dairy Forage Research Center, having some discussion here about doing some feeding trials as part of this work and some estimation of methane production. That may also include some manure management to be determined, but scaling then from the field scale to the whole farm. So how do these decisions we make in terms of forage management ripple through the larger dairy production system? This is a fine root turnover study. And again, another collaboration with Jake and Craig Schaefer. And this is sort of taking it back down to the field scale then, right? So how do these specific alfalfa management practices drive changes in carbon and nitrogen cycling here through fine root turnover? So we've got a pretty intensive program of in-growth coring to measure fine root production, fine root depth, and calculate fine root turnover so we can get a better understanding of total carbon and nitrogen inputs to soil as it relates to things like our fall dormancy selection, harvest management, how frequently are we cutting, and then we also do have some conventional and reduced lignin alfalfa varieties there as well. So thinking about these questions about tissue chemistry as well in terms of carbon and nitrogen cycling. We're in our coming into our third season on that this year. This is another newer project that I'm particularly excited about. We've got a postdoc, Magid Noshi, who's working on this project. This is picking up on one of the legacy projects from some of the previous folks in our unit. So Joanne Lamb and Michael Russell had done some selection on alfalfa for both high and low nitrate uptake. Now they did this with bromide as a proxy for nitrate. And develop these divergent populations. So we've got alfalfas with high fibrous root structure that either have a tendency for high nitrate or low nitrate uptake. And, you know, there's some obvious applications for the high nitrate uptake when we think about remediation and things like that, right? This study, though, we're sort of more interested in the low nitrate uptake, with the hypothesis being that we've got potentially greater nitrogen fixation in these low nitrate uptake alfalfas, right? Alfalfa is a pretty efficient nitrogen scavenger, but if it can't rely on exogenous soil nitrogen, right, will it be fixing more? And then thinking about that in terms of the stability and productivity of alfalfa grass mixtures, right? So this competition for nitrogen comes at a cost for co-planted grasses, right? Alfalfa will outcompete for that nitrogen. So if it's not competing for that nitrogen, it's fixing more of its own. We might be able to design better, you know, more productive alfalfa grass mixtures. So this is sort of a first step in sort of picking that material up and running with it. And Magid is designing a series of both 15N enrichment and then natural abundance studies to sort of look at nitrogen partitioning between the alfalfa and the grass. So we've got that planted last fall, starting up in earnest this spring. And then I guess the final project that I'll mention today is another collaboration. This is with Melissa Wilson, Paolo Pagliari, and then Dan is also doing some work on this, and he'll talk a little bit about his data as well. And this is a study that's kind of focused on, again, sort of broadly thinking about reconnecting nutrient cycles at the farm. So alfalfa typically gets a little manure prior to establishment, but once you've got a perennial established, getting those manure nutrients out is a challenge, right? And so on concentrated animal feeding operations, they end up then with excess manure. They're over applying to silage corn and other annual crops. And so working with Melissa on some of her ideas on more modern manure management technologies, and then also modern alfalfa varieties. So we've got a traffic tolerant alfalfa in this. Oh, look at my images, did get all wonky here. So we've got a conventional high yielding alfalfa and then a traffic tolerant variety. And the mechanism for this traffic tolerance is really typically that they've got broader, more deep set crowns. And so it's really just more resistance to sort of mechanical damage, right? And they were sort of trialed in like grazing systems, and then talking with the seed company, really just kind of just dragging the tractor over it more often. But there hasn't been a whole lot of assessment, you know, sort of in this sort of more research-based context in terms of how those varieties might respond to the increased manure traffic. So we've got a study that we established at Rosemont, and then a couple of years ago, and then we've got a new site going in here at Waseca, and we're looking at a variety of responses as it relates to plant health, stand longevity, yield. Melissa's lab is doing some work looking at ammonia volatilization, so where we deliver that manure surface versus subsurface in terms of how much of that manurial N are we retaining in the system. So I'll show just a little bit of data. This is first year data on crown health. So as we think about alfalfa's persistence and resilience to this increased mechanical disturbance, we did see actually that our traffic tolerant alfalfas had a higher proportion of healthy plants. So this is a crown scoring system that Debby often uses. And so we see under the traffic tolerant variety under manure application actually had slightly higher plants than all the rest, which is an interesting finding. I think that's first year. So again, don't necessarily expect to see a whole lot with especially with the perennial like alfalfa. Often the variability in that the first year data is overwhelming. So this is a good sign. And then in terms of root biomass, we actually also saw an impact of the manure applications as well. And this would be pre-establishment, right? So in first year, we did these assessments were done actually prior to the second set of manure applications, so we don't yet have data on, say, the subsurface versus surface applications of manure. And then just, this would be second year yield, so this past year. A lot of bars here, but in general, we're seeing higher yields with our conventional varieties so far, which is not surprising. It's got a higher yield potential. Where we would really expect the traffic tolerant variety to start shining is in years 3 and 4, right? If it is indeed more resistant to the increased traffic and the mechanical damage associated with these trips across the field, we might see this difference play out in the next couple of years. Interestingly, we, in our conventional alfalfa that received dairy manure prior to establishment, we actually, so we had, I think, higher baseline fertility prior to the second application. And our surface banding and shallow manure injection resulted in reductions in yield, which I think is interesting relative to the broadcast. And I'm not sure exactly why that is. The manure broadcast may just have a better distribution of those nutrients on the surface. But so far our shallow injection, which is really when we think about mitigating things like nitrous oxide emissions and ammonia emissions, maintaining as much of that manure nitrogen on site as possible, that shallow injection provides the best option for that. But we're really not seeing, I should say, we're seeing that's having a pretty detrimental impact on our first and second year alfalfa yields to this point. So
Because of stand destruction.
probably, yeah. We haven't seen that bear out yet necessarily in the Crown Health data, but I would expect it's more, populations.
So over there, can't they?
Well, right, we do have, so that we didn't see any difference in stand counts last spring, but this will be the test this spring, right? Do our populations differ out there? So. But I expect, that's exactly the case. So from here, I think I'll turn it over to Dan and some of the work that he's been doing as it relates to the manure studies.
Just a question in your.
Yeah.
The second tower study with alfalfa,
Mhm.
monoculture versus mixed with grass.
Mhm.
I guess I didn't know there's another tower that you had to work with, so that's cool. That must be the case, right? That there's, okay,
We've got a couple more that we were able to purchase last fiscal year, yes.
okay, cool.
Yeah.
And then also just what's your expectation as far as the feeding studies, methane emissions between monoculture versus mixture?
I don't know if I have a good answer to that. Joe might. We don't have a firm design yet on the feeding study. Ken Kalsher will be leading that at Dairy Forage Research Center. And so there's still a lot of discussion about what that's going to look like, whether or not we'll be able to meet his needs for forage from the field scale study or whether we're going to need some small plots to get more precise like ratios of alfalfa to grass mixtures.
Sure.
My understanding is that with our mixtures, we theoretically could or should end up with lower methane production.
That is correct. There is a difference in methane production between legumes and grasses.
Okay.
So
The legumes, is that lower?
I think the legumes are lower,
Yeah,
right?
that's been the kind of conventional thought at the moment.
Right.
Is that the more efficient the food is.
The lower it is, but that's lower the emissions on the enteric,
For sure.
but not on the back end.
Yeah.
The other issue with methane emissions. Sorry.
Okay, thanks.
Hey, Dan.
All right, so I'm the soil microbiologist of the group, and I'm broadly interested in microbiomes and agroecosystems. So I have a number of projects going on, looking at alfalfa microbiomes, trying to identify core microbiomes, trying to build beneficial microbiomes for inoculants, and trying to identify microbiomes that help plants deal with abiotic stress as well. But for this talk, I thought I would just give a little update on some of the data we've been getting in on the project that Josh was just talking about, which is one of a number of projects we have looking at manure and microbes. So broadly, we want to develop a better understanding of interactions between manure and microbial communities. And this in this particular project, we wanted to look at if different manure application methods impacted soil microbial communities across the soil depth profile. And also determine if manure application impacts alfalfa associated microbiomes in the rhizosphere. So Josh already gave us a nice overview of this experimental design, but I thought I would include a plot map here. Because this becomes important later. So basically up north this way, you'll notice there's two rows of plots, one on the east side and one on the west side here. And so I'll show you a little bit later why that's important, but just note that. So in this experiment, we sampled soils at 3 different depth increments, 0 to 6, 6 to 12, and 12 to 24 inches, about 6 weeks after the manure treatments were applied in the fall. And then we also sampled alfalfa plants and extracted rhizospheres and roots about 8 weeks after the treatments were applied. And from these samples, we just did what I guess is now fairly straightforward paradental sequencing of just bacterial communities so far. And so when we look broadly at what the drivers are of the soil microbiome composition, we found that soil depth naturally is extremely important. So this is a non-metric multidimensional scaling plot where the different colors represent the different soil depths. So there's very clear differences across that depth profile. And what showed up kind of unexpectedly was that the orientation of those plots, the east side versus the west side, were also very, very different, even within that small experiment. So you can see the circles here cluster all on top and the triangles cluster all on the bottom. So this is kind of a not very important, I guess, for the experiment, but sort of a cautionary tale about how variable soil microbiomes can be in sometimes unexpected ways.
There's Rosemont, by the way, so if anybody's familiar with soil variability down on the flats there.
Yeah, but like directly across that 10-foot alley.
Yeah.
That's
Well, I look back at Google Earth to try and see, like, is there anything in recent imagery that would suggest that there should be a difference there, but it was looked like solid blocks of annual crops for at least for the last, 8 to 10 years that we have images for. So there's some strong legacy of something there.
Yeah, which field is it?
This is directly north of our I-10, right next to the Forever Green Field. Plots just north of it. Yeah,
You said for like 8 or 10 years it's been in annuals?
I don't know the exact number, but I went back and it just looked like solid blocks of annual crops for all the recent photos I could find.
So many experiments wiggled into different places.
So just south of there in I-10 though, there was part of the munitions facility, and when you look at our
That
larger
too,
field,
yeah.
there's a lot of old footprints of foundations that
Yeah,
emerge.
I've seen that too.
And
So concrete.
Having worked at Rosemont for 48 years, I'm just.
I bet you.
Yeah, there's some buildings that have been removed. Yeah.
Yeah, you can, especially under drought conditions, our field just south of there, you can see clear as day foundation footprints.
It's probably on top of a bomb.
Yeah, I mean, we've got buried A horizons in that field. It's, not this one that I've seen, but
So that'll make for a nice discussion.
yeah. Right. Surprise.
So, but when we looked at the actual manure treatments, which are mapped to the same NMDS plot in color, basically there were no patterns at all related to the manure. But we wanted to dig into that a little bit deeper. And so we split up the samples by depth increment and use constrained ordinations to partial out that really strong effect of the east side versus the west side. And with this approach, you can see real clear clustering of bacterial communities by the different treatments at each depth. And especially, the shallow injection and the surface banding, which are the green and the blue, tend to be really different from the other treatments. But this was only significant really in the 0 to 6 inch depth increment, so just the top layer.
So are these different communities, the different colors too?
Yeah, so each of those points is community, right? Or the community from a particular soil sample.
So the green dots, are they all from one community or is every single dot a different community?
Those are all different samples from that, in the green case, the surface banding treatment.
Does this relate to your Illumina data or not?
Yes, so each of those points is a community profile based on the Illumina data.
Okay.
So if we, kind of constrain the analysis to look at just the manure treatments within each depth, we can pull out some interesting patterns.
You're gonna talk about what the differences were, right, in terms of the communities.
Yeah.
Yeah.
So to dig into this a little bit deeper, I did some differential abundance analysis using this Ancom BC method to identify some specific bacterial genera that differed in relative abundances between these treatments. And so there were quite a few. So this was just for the 0 to 6 inch depth increment, because that's where we saw those significant interesting differences. There's quite a few genera that are differentially abundant. And this is especially true for the surface banding and the surface broadcast treatments. You can see in the heat map, there's a lot of taxa that are more abundant with those treatments. And what I thought was interesting is that many of these genera aren't typically very abundant in soil. So you get things like Mombutsia or Vanilla bacillus that you never really see in soil. And these are probably just coming directly from the manure itself. So those seem to be surviving in this case in soil.
What's their function?
Yeah, that's a good question. For a lot of these, we don't really know their function. These are probably coming from the gut or surviving in the raw or sort of in the soil.
This Pseudomonas one, is that like Pseudomonas syringae?
So I mean, Pseudomonas is a really diverse genus.
Okay.
That's probably not syringae,
Okay,
but it could be stuff from the gut, or it could be just natural soil populations that respond to or are activated by the manure and the influx of carbon.
all right, yeah, I'm just
So-
curious, because that's been problem for like some of the disease we see in the oilseeds.
Oh, yeah.
It's, yeah,
Yeah, Debbie's probably the expert on the foliar pathogens.
Probably coming from the manure that was applied in your plant.
yeah.
And we nailed it.
So we also look at alpha diversity, since a lot of studies suggest that manure applications improve the diversity of soils. But we didn't really see that here. We saw the strong impact of soil depth. Which is pretty typical as you go deeper in the soil. There's communities are less and less diverse. That typically tends to track soil pH or organic matter gradients. But really no impact of the manure treatments on diversity. And so switching up a little bit, when we looked at the rhizosphere communities of alfalfa in these plots, to determine if manure is driving changes in these microbiomes at the kind of the plant soil interface. Again, we saw this real strong effect of the plants growing on the east side versus the west side of that experiment. So there's no real buffering by the plant or selecting for particular communities there. We saw a weak effect of the different high traffic standard varieties, but I think that might be because some of them tended to be found more often on one side of the field. And again, there was no obvious treatment effect of the manure on the alfalfa microbiomes here. But again, when we use these constrained ordinations to delve into this a little further, There was a significant effect of the manure treatments. But then we lost that difference between the different alfalfa varieties. And there's a marginally significant treatment by variety interaction, which I think could potentially be interesting as we collect more data. So in this plot, you see the normal fertilizer and the non-treatment without anything tend to cluster together, and all the manure treatments tend to cluster together. But then also you have the regular varieties here, the triangles, which tend to be separate from the high traffic tolerance varieties. So
There's that whole other issue is, are these relationships repeatable?
right, yeah, we'll find out, I guess.
That's what I mean, that's, even those of us that have dabbled in this a little bit,
This is 2020,
This is the first year of data,
2021.
so we're still waiting on the second year.
Right, right. So this is only after 1 year of establishment of the alfalfa first year sampling. So,
Yeah,
that's not even what I'm thinking about another site.
well, right.
yeah. Yeah, it gets hard.
How many reps, how many reps, you have?
3 reps.
There's 3 reps, but within each of these,
Yeah, field reps, I should say.
yeah, I think we have 10 or 12, I forget. I can't count from here.
Yeah, I wonder that correlation, repeatability between reps or the CV between reps, etc.
Yeah, so rep had the block anyway had a pretty small effect. But as you can see here, I mean, the treatments are fairly consistent. Again, when we look for specific bacterial genera that differ between the treatments. And again, the surface banding and the broadcasting enriched for the alfalfa microbiome for some of these genera. Again, some of these are interesting, like the Rhambutia, the Acinetobacter, that you might not necessarily expect to find at high abundances in soil. So, there's potentially manure-borne microbes that are colonizing the alfalfa rhizosphere.
Did you measure your micro populations in every plant before you put a treatment on it?
We did not do that, I don't think, in this
You know what I mean?
experiment. I don't know if
To
those.
know that variation and what you started with, because of the microbial variation that you had, we all know, right?
Right. And I don't know if those samples exist, like the pre.
You know, what I mean, doing in
We'll have
sampling
dried samples pre-establishment, but we didn't.
Okay.
in all plots. I don't mean general sample, I mean. In each damn tree.
Yes, we do. Yeah,
Yeah.
we don't have them frozen field moist.
Okay.
This was before Dan. was
Yeah,
involved.
so
What are,
with the second experiment that's getting set up, I think we do have those.
Yeah, we've got another site going in this year.
Based on some of the work we've done with Sadowski and these cropping system things, that damn variation, it's.
Yeah.
Yeah.
Ooh.
would it be helpful? Would it be helpful?
You know what I mean?
Yeah, you go someplace else.
Oh,
and it's
my.
totally different.
Would it be helpful to analyze any of our samples from the EddyFlux project? Because we have frozen samples. We have samples that might be seen from that too.
Maybe, I mean, it's close, but it is a different field, so and as we saw, the legacy effect just in this field is pretty
Yeah.
substantial. So I don't know.
I guess if.
Knowing where you started
Yeah.
Yeah,
in each damn plot.
yeah, we have samples for each plot prior to treatment, but
Yeah.
it was before Dan was on the project, so they're all dried down, so for microbial analysis, it's
Yeah, you
not
can't do it, yeah.
useful.
Yeah.
Can you
well,
still get DNA from it though, or no?
No,
You
it's
can't.
I mean, you can get DNA from it, but it's not
not good,
useful.
yeah.
Because if they're just sitting there dried, things change.
Even if there've been like any number of method studies, you probably know them better than I do, but like even at -20, like.
Yeah.
Like if you don't store them holding up,
Mhm.
you also get,
Right,
you
Yeah.
start to get changes, because there are bugs in there that can cope with -20, not -20. But if anyone was interested in chasing this variability question, yeah, I have samples that I might see from that other project.
I have a quick broader question. So when you first started, I thought there was gonna be something about like the bacteria and the nodules or something like. How does this relate to alfalfa? And like, what's the practical? Is there like, what's the practical of the different communities? Does it relate to not, it doesn't necessarily relate to nodulation, but what else?
Not necessarily inoculation, I mean, just the rise of bacterial communities plays a big role in plant health, disease suppression, nutrient acquisition. Things like that. It's hard to infer those functions from marker gene data. But this is a fairly high throughput way to study those communities and how they respond to management practices, right? Okay. So,
Can you dig a little deeper and try to identify species that are beneficial versus not, or is that the data is just not robust enough?
yeah, I mean, that's hard to do with marker gene data because it's such a short region of the 16S. It's usually not great for inferring function. There's some methods that try to do that, but I don't believe them. You'd probably want to go in with shotgun metagenomics and try to reconstruct genomes.
Especially if we're considering across trials, across locations, that may be the best approach, or you
Right.
have, billions of leads.
Yeah, I mean, you probably need 50 to 100, 200 million reads per sample to do a good job, which is expensive.
I need to
How-
run, but I'm chatting with you guys too.
I'm about done here, but so basically, manure impacts on soil microbiome were tend to be limited to the top 6 inches depth increment. Some of those manure associated taxa might be carrying over to the alfalfa rhizosphere, but we don't really know if these are beneficials or pathogens. What I thought was interesting was that the shallow injection appeared to have less of an impact on the soil and rhizosphere communities than did the banding and the broadcasting treatments. Which is kind of opposite than I would expect. But maybe the shallow injection is just mixing those in with soil environment better, exposing those microbes to different conditions and competition with indigenous microbes. So I'll be curious to see in the future, in these subsequent years, is this repeatable? Does the additional application of manure drive further changes into soil communities? And we'd also like to incorporate some analyses of fungal communities as well. It'd be interesting to see if with those traffic tolerant varieties that are maybe less injured by traffic, will they be less susceptible to potential fungal pathogen colonization? So that's all I had.
Dan, how much soil sample needed from like one plot or one location? And the reason I'm asking, so as you may know, we planned 2 experiments for the gene editing to test the phosphor super uptake. We planted in 2 locations that 2 miles away in Rosemont. Each location has 0.14 acre, really small. And I wonder if you do not need a much sample, whether we can test the microbial changes, or if any, under gene edited or non-gene edited, high super uptake, hyper phosphor uptake, and compare with the checks, etc.
Yeah, that might be interesting. You don't need very much soil for DNA work, at least not for this marker gene DNA work. Usually you only need like 1/4 gram, and you can do with a lot less if you need. For that work, I'd be curious about AMF in particular and sort of interactions between the high phosphorus uptake and potential AMF communities.
Yes, yeah. That would be great, because no matter what, we will plan experiments this year in the open field, and eager to know whether they have some impacts on the microbes.
I don't, know how to look at the chat. Down in the presentation.
Yeah.
Chat.
It is, but I can't get to it.
Your cursor's just over on there, so this got a little left. There you go. Oh, this is the paper that Jerry Cohen posted a while back.
Yeah, and you were talking about looking at one by next. Have you looked at greenhouse?
We have not.
Are you aware of Adria Fernandez's paper?
I
With Sadowski?
I think I've seen it, Schaefer.
Have you looked at it?
I haven't looked at it recently.
Well, I would encourage you to look at it because it was very frustrating. And I feel, if I listen to you, that same level of frustration is there and sorting through this. You know, when we had a whole series of inputs, including manure, fertilizer types and all of that stuff. And those were not the major drivers of the microbial populations or the functions within those microbial populations. You know what it came down to?
pH.
pH and soil type.
Yeah.
That variation that you're kind of talking about. Bam, they're popped up again, right?
Yeah, there's major drivers
Like
that.
major
but it-
drivers that swamp out those. Yeah, so. That's a really. Thinking strategically about this type of work, I mean, I found it. Working with one of the best guys in the country, right, Sadowski, I mean, in terms of trying to work through that, it was really frustrating. But that's been a few years ago, I'm sure things have advanced.
Oh, yeah, now we're all more frustrated.
With the whole idea, right, you know, in terms of, you know, it's interesting to see those differences in the populations, but function.
Yeah,
Getting a handle on function. And in case of your manure treatment, what happens to those microbes that came from the manure, that came from the livestock, what happens to those? Are they important in terms of a function, you know, in that system over time? Right?
yeah, that's hard to do.
And
We're
if you
getting
look-
better at it.
Yeah, and if you look at the carbon sequestration information coming out of that long-term cropping system trial in Wisconsin, right, where the manure was added back to a pasture system. It was in fact, in those where the carbon capture was real, right? The other ones, you know, I'm convinced or not, but that was one wasn't. I always wondered what the relationship was in terms of not just the organic matter, but maybe the microbial populations that were being put back into the system. That popped again today when you were talking about the residence time.
Yeah,
You know, some of those microbes that were being put out there that weren't there originally.
yeah, we've done some microcosm experiments as well that were
Yeah.
initiated by Josh and a former postdoc.
Yeah,
And from those, it looks like most of the manure-borne stuff just dies off.
I think
30 to 60 days is what we.
Yeah,
30 to 60 days. That's, yeah, so you already know.
So newborn stuff dies off. What about the indigenous microbes? Do they get primed and changed and they come out the other side differently than what they went in?
yeah, that's kind of the story that we saw in that.
Okay, because that could be the more impactful.
Right,
Correct.
yeah, that was part of the reason I was interested in looking at that.
And it could be a nothing burger too.
You know, it's the manure priming some of those copiotrophic bacteria that can then go over and colonize the alfalfa rhizosphere, survive there.
Interesting. Jerry?
So I feel like I'm being the naturalist today, but so since we're ending on microbiology, so the early start, the question was a lot of the digestibility of alfalfa, especially for pentose metabolism. And so something I picked up years ago when I was working at the NSF and we were looking at microbial ecology is that some ruminants, mainly moose, for example, actually change diet in a rather dramatic way as they migrate over the winter and summer. And they end up in a high pentose diet. And there's a microbe that's not culturable. I think it's called BS11 in moose, which actually will ferment xylose and ravenose. and other pentoses. So the question is, would it be possible to change cows? One ruminant must be similar to the other, and look at that in terms of digestibility of alfalfa in a ruminant environment that might be altered in a way that, because cows don't actually migrate in summer and winter as much as they'd like to. But things like moose do, and they do change their diet, and they change it dramatically. I just throw it out because it's there's a lot has been done on pentose metabolism for biofuels, so we know quite a lot about it. But in ruminants, the best ones actually look at wild ruminant microbiology. I know you only want to work on manure, but I think there might be a lot of interesting culturable bacteria in ruminant metabolism that could be useful.
Yeah, the Dairy Forage Center, they have people doing similar sort of work, looking at rumen microbes, and again, they vary from cow to cow and feed and all kinds of things. So yeah,
But moose are a lot more fun.
Jerry, I'm going for goats, because goats are a lot of fun.
Yeah, but goats don't have to migrate, so. I mean,
Oh.
I think that I think the deal with the moose is that it has to have this high pentose diet during part of the year, and that's, at least that's.
Mountain goats, Dr. Cohen, do migrate.
What's that? Some goats aren't supposed to migrate.
Mountain goats do migrate up and down the mountain.
Oh, that's
Right.
true. Yeah. The wild ones, that'd be true.
We know that altitude is important for plant growth.
Yeah.
So,
Well, anyway, I just throw that out because it's just one that hasn't been put on the table, but since you end with a microbiologist, I figure that at least start with that. I will put in chat paper that relates to that. If I can find out how to do that.
So we've probably lost a lot of people online too, but if there are suggestions for how we can start to organize a supply chain group for alfalfa protein, I'd like to have your comments on that.
Well, comments, we would, our team would be very willing to sit down and not just have but actually put a strategy together. And we've talked before in terms of, just like we've done with all of the other crops. Basically it doesn't happen overnight.
Right.
It's basically keeping a team in place for a long enough period of time that can explore the possibilities and relate that back to the development programs. So you're investing, we are investing in a way that brings those potential components of these new crops or old crops or modified crops into something that there is an emerging demand or discovery of a demand that isn't there, right? And help develop it as you guys with pharmaceutical work and natural products, a whole array of things. And you never know. You never know during that discovery process when you're going to find it and what it's going to be, right? That's what we learned out over time. All want someone that is out there that has been waiting for a contact with a new idea, and then these things develop. We can give you example after example of that. So having a team that's steady and in place and in communication on a repeating basis is what I would suggest. Not just a conversation, but actually have a team that's part of the overall commercialization effort for evergreen or crops in general. To have a steady conversation and people that are looking out for those connections as they talk to various organizations.
Yeah, that's why I kind of want to have a big kickoff meeting of some sort to bring all kinds of people together and find people to form that team who are excited.
Well, I've been here a long time, and that story you told, my generation was part of that work. Right, not directly, but indirectly, the individual that came to you we held a big damn symposium here. College-wide symposium on this issue.
Wow.
In terms of how do you extract protein, and that time the big deal was juicing it. Right? That piece of equipment was juicing it. But there was also a strong component and support for it in Madison. USDA group in Madison
Right.
had 3 people, 2 or 3 people that were tied to that program. So it was a really good network of activity, and then like you described, for a whole series of reasons. You know, people maintain interest, but IH, you know, got moved around and their whole structure changed, and that equipment actually got turned into scrap metal. Right, they called and said, I called over and said, where's that equipment? And it was just that, a month before they had just scrapped it. And yeah, Kent Crookston, you remember Kent?
Yeah.
They tried to hire him to run that program.
Yeah, follow-up question, that is, well, you are the for our green director, what kind of results are you looking for from the research area? Then what kind of preliminary results you see, oh, this is the direction we should go. And for our green, we'll need to fund it for a special or specific goal. So what kind of preliminary
I was
results
hoping
you are
that
looking for?
USDA would put major funds into the Forever Green effort. You know, so, well, this is what I would say.
Well, you have 6 scientists, so.
Not bad.
You know it.
Yeah,
Is what I'm saying in terms of, but to make an investment, we all have to agree what the investment would be to move towards a product. And as a director, co-director of Forever Green, I don't have that, but I know how to get it. Right, and the connections that I would make, you guys have your food group, your food processing group. They are major players. We have major players here. We have the Plant Protein Innovation Center here on campus. We have the entire food science department. And out of all of that, we also have connections to General Mills and a wide range of other food companies. So to me, they need to hear about this team and the possibilities and the capacity this team has with and connected to the USDA network of scientists that can provide the base support for moving plant materials and ingredients out into end uses. They're not going to raise their hands at you. No, it has to be a discovery. Right? And most of them would not have thought the food industry. I keep bringing it up. They would never have thought. Most of them would not have thought about alfalfa protein for direct human consumption. So that those connections and those relationships have to be built. There has to be feedback to the development team as to what are the potential forms of that protein. And as you guys know, the initial pushback was in the initial harvesting of alfalfa, it's got a bunch of damn amino acids, right? They want functional proteins. I know that they want functional proteins. So figuring out how to put into the marketplace functional proteins for incorporation into food products, that would be the working relationship with the food industry, in my opinion. But you have all the players here. You have your players in USDA, but we also have the stateside players right here on campus with those connections to the food industry that are readily available. Are you guys a member of EPIC?
no, we should be.
Yeah, you should be. That's what I'm just saying. Get into that hopper of discussion through the Plant Protein Innovation Center with Pam Ismail. Have you guys met with Pam?
Yeah, Pam and I are working
All
on
right,
plant protein right now.
there you go. That would be, for me, would be the avenue.
But she has a lot of projects on her
She has a
plate,
lot of projects.
so.
I know, but we always help her,
Yeah.
right? And that's where the funding comes into play, right? That's the game. We have the USDA scientists funded. We have the food scientists funded, they're engaged. Now, how do we build capacity? All of us have historically have a lot of things going on, but hiring, for example, a short-term research assistant professor. Right, that would work on behalf of the entire team. In some area that would be necessary to move program forward in that direction. Those are the types of things that need to be thought through. What type of investments are really needed? And most of you know me well enough. I'm not a project guy. I'm a program guy, right? If we're gonna put a program together that's moving in this direction and laying it out in terms of the strategy. That would enhance the planting of this pruning on the landscape. I think it could very well fall within. I'm not the decision maker in funding, but a great argument could be made for that investment if a strategy was laid out.
Our artwork is getting bigger for sure. We work with medicine. The person over there will study the proteins to how can we keep the protein stable when we harvest them. And with Peoria, 2 scientists. So the USDA efforts get bigger for sure. And that's good news.
But for me, starting well, just talking about funding, there has to be a strategy to figure out. And if we're just looking at funding from Forever Green, the Forever Green funding is designed for initiation, right? And contributing to a strategy that's being developed, right? And I like to see, and as you guys all know, the Forever Green platforms have a strategy. Each team has a strategy. Here we come with Alfalfa, paying more attention to it from the program. I would love to see that strategy and see where state resources would fit into that strategy and be useful in combination with the federal resources.
Yeah, me too.
Yeah. Yeah. So it isn't any different in this case than any other case, right? In terms of whether it's Kearns program has a strategy, it's obvious where investments need to be made. In Alfalfa, I don't know all of the network of the USDA, what capacity is available within the USDA. But obviously, I've told you for 20 years that the team is very interested in trying to think about how we get more alfalfa out of the landscape and looking for the new avenues of making those planting possible.
Right, well, let's wrap up the meeting and we
Yeah.
can keep chatting about.
Yeah.
How to go forward on that, so, thanks, everybody, bye.
Thank you so much.
Bye-bye, thanks, everybody.